Hi everyone, we are Group 11. In this video, we'll talk about Ig class switching. Before we get to the main point, please let me talk about immune system. Immune system is a system which our bodies recognize pathogens and protect you against viruses and bacteria. How does the immune system work? Let's take the example of a bacterial invasion of epithelial cells. First, when bacteria invades our bodies, macrophages and neutrophils phagocytose bacteria immediately. This is the innate immune system. If phagocytosis is insufficient, the acquired immune system becomes more active. Langerhans cells, which have engulfed bacteria, move to lymph nodes through lymphatic vessels. In lymph nodes, Langerhans cells mature into dendritic cells. After that, they present information about the bacteria on their membrane surface. This presenting process is known as the antigen presentation. Second, helper T cells are activated by this antigen presentation. Helper T cell is a type of T cells which responds to antigen presentation and produces some substances, which is called cytokines. Cytokines enable B cells to become plasma cells. And they produce antibodies specific to the antigen. Next, antibodies are transported to the site of bacterial invasion along with blood flow. By binding to the specific antigen of the bacteria, phagocytosis of neutrophils are enhanced. This effect of antibodies is called opsonization. Here's a question. It is said that humans need antibodies corresponding to 1 billion. To produce such a variety of antibodies, 2 trillion base pairs of genes would be required. But the human genome only has 3 billion base pairs. How does the body produce such a diverse range of antibodies? The concept discovered to address this is Ig class switching and gene rearrangement. First, let's look at the basic molecular structure of antibodies called immunoglobulins. The longer chain is the H chain, and the shorter one is the L chain. The orange region is the variable domain, and the blue region is the constant domain. Immunoglobulins are classified into 5 classes based on differences in the constant domain. IgM is initially expressed in B cells, so how does it differentiate into the 5 classes? The answer lies in the concept of Ig class switching. For example, when activated by cytokines to produce IgG, the constant region coding for the blue part of the antibody is removed, including C-mu and C-delta. This allows C-gamma to come right next to the gene coding for the variable region, enabling class switching from IgM to IgG. IgM primarily forms pentamers, aiding in efficient bacterial aggregation. IgG constitutes over 70% of immunoglobulins and plays a role in neutralizing bacteria and toxins and opsonization of phagocytes. Next is a mechanism supporting antibody diversity, gene rearrangement. The V, D, and J genes, which code for the variable region, each contain about 50, 30, and 6 genes, respectively. Choosing one from each category allows the production of 9,000 different antibodies. The same mechanism exists for the L chain, and by combining the H and L chains, the body can generate as many as 1 million different antibodies. Cytokines play a crucial role in class switching. Cytokines are produced by follicular helper T cells, and the antibodies that switch depend on the type of cytokine. The cytokine IL-4 controls class switching to IgG1 and IgE, IFN-gamma to IgG2, and TGF-beta to IgA. In the process of class switching, AID, activation-induced histidine deaminase, serves as an essential enzyme. AID is expressed exclusively in activated B cells and induces cleavage of the constant regions of antibodies, as well as rearrangement of DNA. Upon receiving signals from different cytokines, class switching to IgG, IgE, and IgA occurs. Therefore, in the case of AID deficiency, the produced antibodies are limited. leading to immunodeficiency. In this way, Ig class switching is crucial for producing a diverse range of antibodies in the immune system. I'd be delighted if you gained even a bit of understanding from this video. Thank you.